Hi everyone, in our previous video we looked into type 1 regulatory T-cell mediated immune regulation in malaria, and I think we got up to the point where we looked into the 4 major mechanisms employed by type 1 regulatory T-cells to control immune response, and they were mechanisms that were illustrated in these panels right here, panels A to D. In this video we're going to focus on the mechanism outlined in panel A specifically, which is cytokine mediated mechanisms. So basically, in this specific type of mechanism, these type 1 regulatory T cells, they release cytokines such as IL-10, and they also release transforming growth factor beta. And these just reduce the activity of antigen-presenting cells, also known as APCs. Now, these antigen-presenting cells are immune cells that show fragments of the parasite. They're immune cells that essentially show antigens or fragments of the parasite. To T cells as means of initiating an immune response. So by down-regulating APC activity, these type 1 regulatory T cells decrease the expression of MHC class II molecules and other co-stimulatory signals that are needed to fully activate these T cells. As a result, we have fewer inflammatory molecules such as IL-2, IF and gamma, or GM-CSF that are produced, and this essentially just keeps inflammation under control. Now, one thing that you might have a question about whilst looking at this diagram could be these molecules. What are TH cells? Well, these TH cells are T helper cells. And these T helper cells are basically a subtype of the CD4 plus T lymphocytes that we looked at in our earlier videos. And their job is to essentially just coordinate the immune response by releasing cytokines. And because these cells drive inflammation and help activate so many of the other immune cells, too much of the T helper cell activity can cause autoimmune diseases and chronic inflammation. And that is why these type 1 regulatory T cells exist. They exist in order to prevent excessive T helper cell activity. And the way that these type 1 regulatory T cells does this is by, as I mentioned earlier, using these molecules, producing these molecules, but specifically producing the cytokine, producing high amounts of IL-10. And this is very powerful anti-inflammatory cytokine in the immune system, so I think it's very important to go over its role here. It's a very powerful anti-inflammatory cytokine. Now, IL-10 essentially has 3 major roles in kind of doing the job of an anti-inflammatory cytokine. It firstly shuts down or decreases the T helper cell cytokine production. And so basically, these T helper cells stop producing IFN gamma, IL-17, IL-4. IL-10 basically shuts down these cytokines produced from T helper cells. And it also reprograms the APCs into tolerogenic APCs. So basically, these APCs will now present less antigens, they release less inflammatory cytokines, and they cannot activate these TH cells, these T helper cells as well. Also, another thing is IL-10 essentially reduces the proliferation of T helper cells, so it just essentially stops the expansion of effector T cell population. the disease of malaria, the specific mechanism just prevents excessive immune activation in organs such as the liver and the spleen, which can otherwise lead to immune-mediated anemia and tissue damage as well. Additionally, these IL-10 cytokines, they promote the development of regulatory APCs. We can see that in this diagram right here. Regulatory APCs, which is a type of APCs, a common form of APCs that produce more IL-10 and fewer pro-inflammatory signals, and this essentially just helps to restore immune homeostasis. So that's pretty much it of cytokine-mediated mechanisms. In our next video, we'll be looking at cell contact-dependent mechanisms.